This presentation will describe a new web-based tool to group genes by their expression profile. A gene expression profile shows the mRNA concentration levels that exist in a cell or tissue. This measure is done with the help of microarray chips. One microarray can measure the concentration levels of thousands of previously known mRNA chains. In the figure on the right, each column is the output of a microarray and each row shows the expression level of a probe set in different microarrays. A probe set is a collection of probes designed to interrogate a given mRNA sequence. Some probe sets can match different sequences of the same gene. Others can match a sequence that appears in more than one gene. We are interested in finding genes or probe sets that show a similar variation of its expression on different external conditions. A gene will be underexpressed under certain condition if it presents a lower expression level for that condition. In a similar way, a gene will be overexpressed if it presents a higher expression level for a given condition. There are many algorithms that can be used to group genes that show similar level of expressions. In this presentation we will focus on a new B-clustering algorithm called K-formal concept analysis. Formal concept analysis group objects that share some common attributes. It is possible to represent these formal concepts in a lattice. The lattice on the right has a diagram of the table presented on the left. The colored boxes on each side represent the same concepts. K-formal concept analysis is a generalization of formal concept analysis. It starts with an exploration that sweeps over all possible thresholds in the max plus and min plus semirings. Each threshold will give origin to one lattice. The max plus semiring will group the underexpressed genes and the min plus the overexpressed. We have created a web page that shows how this B-clustering algorithm works. This is the workflow of an analysis. First, the data for each experiment is uploaded to the web, and then some preprocessing is done in order to feed the raw data into the clustering algorithm. After this step, the K-formal concept analysis algorithm is run, and each of the concepts produced at the output will represent a cluster. The input data will be a .cell Affymetrix file. Currently, the web supports these 4 microarrays. At the Gene Expression Omnibus database there are thousands of datasets. We will search for an experiment which contains the gene expression of roots and shoots from Arabidopsis thaliana grown on selenite. There are 8 cell files which can be downloaded here. Once we have the files, we can go to the web page and start the analysis. First, we must log in to upload the new data. Now click on Upload New Data and the new web view will appear. Here you can select the name of the dataset, give a brief description and select the proper Affymetrix microarray chip. Then you can upload the files and click Next when finished. In this example, we will upload the 8 files that we downloaded a moment ago. Now we must select a preprocessor to apply to the raw data. First, we enter the name that we want to give to this preprocessor. Then we select among the 4 different types of normalization. For this example, we choose the default, the arithmetic mean. Then we write a descriptive name associated with each uploaded file. These 8 names are the 8 different cases that we read from the Gene Expressions Omnibus web. There are 4 files for the root tissues, 2 for control and 2 with selenium, and there are other 4 shoot tissues, 2 control and 2 with selenium. This view shows the preprocessor details. The first line shows the name we gave it previously. Below is the preprocessor type. This means that we divide each probe set by its mean value. The data size indicates the dimension of the matrix uploaded, the number of probe sets and the number of experiments. Below we can see the representation of the expression level, which represents the input for the K-formal concept analysis algorithm. We can access by clicking on Explore. The results appear in this view. The first graph shows the number of concepts in function of the threshold for the max plus and min plus semirings. Scrolling down we can see the concept lattice. The slider on the left fixes a threshold and the corresponding conceptual lattice is plotted on the canvas. We will start in the max plus domain, which corresponds to underexpressed genes, and the minimum threshold. We can see that there are only 3 circles. Each circle is a formal concept with a radius proportional to the number of propsets it has. The top concept corresponds to the objects that do not have any attribute. And by contrast, the bottom formal concept has all the attributes. If we change the threshold, new concepts appear. Three concepts deserve a special mention in this lattice. This concept has the propsets that are underexpressed in the root when the selenium is present. If we select one propset, a brief description will appear on the right. In this description there are links to other web resources like Gene Ontology, where more information can be consulted. Other two interesting concepts are the corresponding to the propsets underexpressed in the root, no matter if selenium were present or not, and the other is the corresponding to the propsets in the shoots. We can change the threshold and see how the concepts change the number of propsets they have. This has been a brief description of a GenB clustering tool. You can find more information on this topic in the following references.